Good morning, fans of public-supported science, and welcome to the BioEPIC Slam. I'm Mingfei Chen from Lawrence Berkeley National Lab, also known as Berkeley Lab, and Enigma SFA, or Science Focus Area, which you will hear more about later. Today, we're coming to you live from Berkeley Lab in the beautiful hills overlooking San Francisco Bay and the Golden Gate Bridge. This is where we bring science solutions to the world. We're gathered this morning to be regaled with concise science stories from some of the early career scientists, soon to be housed in what will be the newest building at Berkeley Lab. I will be your host today, and I'll be joined by another one of the Berkeley Lab early career staff with a passion for communicating science. Coming to us from the ESA communication team, he captures science stories from a visual angle using photography and illustration. Please welcome Jeremy Snyder.
Hi, everyone, and thank you, Mingfei. I'm really excited to be supporting this event with you. I think these events are really important as a part of increasing the visibility of our science and giving the chance for our scientists to practice communicating their work in a really engaging way, which I know you are no stranger to as the winner of last year's BioEPIC Slam with your talk about a subterranean society of microbial biofilms. I still remember that talk being super interesting and exciting, and I can't wait to see what this year's speakers have in store for us. So today we'll be bringing you lightning presentations from talented early career scientists representing our 4 science focus areas, or SFAs, that are currently scattered across the Berkeley Lab campus, but which will all soon be brought together under one roof in the BioEPIC building. And for those that don't know, BioEPIC stands for the Biological and Environmental Program Integration Center. When it's completely finished, hopefully later this year, it will be Berkeley Lab's newest interdisciplinary home for scientists from biosciences and Earth and Environmental Sciences area, or ESA, to collaborate on the kinds of research you're about to hear about. And with that, it's my great pleasure to have Paul Adams, our Associate Laboratory Director for Biosciences, and Alastair Rogers, Division Director for the Climate and Ecosystem Sciences Division in ESA, with us today to give opening remarks for our events. Paul and Alastair, please take it away.
Great. Thanks, Jeremy, and thanks, Mingfei. Welcome, everyone. It's great to be here. I'm very excited that we're holding this event, which is part of our journey towards occupying the new BioEPIC building early next year, so it's not far away. As the ALD for Biosciences, I look forward to the many research opportunities that will result from co-location of our 4 Berkeley Lab-led science focus areas, which you're going to hear from today. Going from microbes to ecosystems, BioEPIC will allow us to do something new, which is really to sense, study, and simulate biological environmental interactions across multiple scales. So that's fantastic science that will come from that. As the co-leader of one of the SFAs, Enigma, I'm particularly looking forward to the ability of our scientists to share their ideas with our other experts in the building and increase our collaborations to solve important problems. I'm sure that locating our collective expertise and capabilities under one roof is really going to usher in a new era of discovery for the SFAs. And it's going to help support DOE research, the DOE mission. So this is it's a great day. It's great to hear from all of you. The Slam is always a fantastic event. I'm happy to be here. And I'm going to pass it on to Alastair to say a few words.
Thanks, Paul. I'm pleased to be representing the Earth and Environmental Sciences area today and excited for my first Slam. It's an exciting time for me to join the laboratory as this new facility is nearing completion. And I'm honored and humbled to have the opportunity to work with the talented researchers of both ESA and Biosciences. It's my hope that the potential synergies that are possible at the interfaces of these tenant SFAs can be realized and enabled by the proximity of so many people working together in the same place. The Slam's an important step towards those future collaborations because it's an opportunity to learn about each other's work. Collaborating across disciplines is challenging. The acronyms are different. We just heard from Paul about the scales being different. The journals we publish in are different. A key step to understanding each other is recognizing how much knowledge we all take for granted and how best to communicate in a way that helps others understand and appreciate the amazing science that's happening in these SFAs. Collaboration starts with this shared understanding, so I'm equally excited to see the approaches to communication that you'll all take today. When I think about the national and global challenges facing society, it's clear that our 2 areas can make important contributions. It's going to be an exciting decade, and BioEPIC's poised to make a major impact. Thank you.
Awesome. Thank you so much, Paul and Alistair. Now, here in our virtual room to tell you more about BioEPIC are the two building leads, Susanna Tringe and Peter Nico. Susanna is the director of the Environmental Genomics and Systems Biology Division in the Biosciences area, and Peter is the deputy director of the Energy Geosciences Division in the Earth and Environmental Sciences area. Susanna and Peter, please take it away.
All right, thanks. Yeah, like Jeremy said, I am Susanna Tringe. I'm Division Director of the Environmental Genomics and Systems Biology Division, and I have been helping to oversee the science directions of this exciting Biological and Environmental Program Integration Center, which, as the others have said, is a 73,000 square foot facility that's going to house research supported by the Department of Energy's Office of Science, Biological and Environmental Research Program, spanning both the biosciences area and the earth and environmental sciences area at the lab, meaning it will span scales all the way from the microbial to the landscape scale. It's supported by some really exciting and unique capabilities that you're going to hear more about today from our exciting SLAM participants. And so my colleague Peter is going to give you a little sneak peek of the inside of the building and what's in there.
So thank you very much, everybody, and very pleased to be here. As everybody said, BioEPIC has been a multi-year development scale from several years ago when it was a science concept sponsored by the laboratory to early drawings by architects to finally breaking ground. And now we're almost complete. And you can see that the right-hand side, we're showing the building as it really exists, which is slightly different than the drawings behind us, but really quite similar. And we're almost getting to the point where we're ready to move in, as was said. The outside of the building is almost completely done. The insides, you're seeing a picture here of the new labs. They're still having some things being put in and finished up, but we're starting to take people through the building, take tours. People who are coming through the building are seeing the new lab facilities, the new conference room facilities, the new places where we can all do the work that we want to do and collaborate on the science that we're about to hear about. So we're getting very close to being ready to really move our science into the building and really reap all the rewards that we've been building on for years. And with that, I'll pass it back and we can get on with the science talks. Thank you very much.
Great. Thank you so much for that intro to the BioEPIC facility. Now I'll pass it back to Mingfei.
Now, here is what you need to know about the BioEPIC Slam. You will hear from leaders of 4 science focus area or SFAs. Those leaders will introduce 1 or 2 researchers who will seek to encapsulate complex science stories into a compressed 3-minute format, in understandable language, of course. But you, the audience, will be the judge of that. Be sure to also show your support and love by dropping the applause reactions in the Zoom. You will each have an opportunity to cast your vote at the end for your favorite BioEPIC Slammer. And through the magic of computing science, we'll identify honorable mention, first runner-up, and champion of the BioEPIC Slam. We promise you it will be truly epic. Actually, let's call it BioEPIC. And guess what? Our presenters today aren't just going to bask in your applause through the chats. They're also getting some cool cash. But let me tell you, every one of the science storytellers you're about to hear from is a winner in their own way. Jeremy, why don't you tell us a bit about who's involved in BioEPIC?
Certainly. So let's meet the 4 science-focused areas that will be BioEPIC's anchor tenants, whose leaders will be introducing both their SFAs and our SLAM contestants. We're going to start off with Peter Nico, representing the Belowground Biogeochemistry SFA. Peter Nico is a task lead for the Belowground Biogeochemistry, which focuses on, for task 3, which focuses on organic geochemical controls on soil organic matter cycling. Peter's research is focused on soil chemistry, specifically looking at carbon and how it's stored in soil. He and his fellow researchers use the ALS and other X-ray techniques to understand the chemistry of carbon in soil, understanding how it changes, what types of minerals it associates with, and what makes it more or less stable. So please welcome Peter Nico.
Thank you, Jeremy. And as was said, I'm going to just show an overview of our Belowground Biogeochemistry SFA, which is Science Focus Area, which is a large multi-investigator project. And it's really focused at 2 different locations around California, the Blodgett Experimental Forest in the Sierras and the UC Point Reyes Field Station on the coast. This is a large field-based project which is really about warming a block of soil. You see a picture there and an artist rendering of putting heaters into a multi-meter diameter ring of soil and warming it up to see how things are going to change in a future warming climate. And the result is that we get temperature profiles of our soil and control soil that show the same seasonal fluctuations up and down, like in the winter and the summer, but one set of soil is just a few degrees offset from the other set, so we can compare what warming will do to our soil carbon cycling. The overall project is really tasked with the idea of understanding what the role of soils into the terrestrial feedbacks, carbon going back to the atmosphere, is going to be under changing climate over the next 100 years. And in the spirit of BioEPIC and the integration of biology and earth sciences and multi-scale interactions, we're really doing this in a way where we're trying to understand how the geochemistry, the microbiology, the plant's roots interfaces with the soil and with the atmosphere, how all of those things are coming together to generate the responses we're seeing in our field-based system. And one of the key young investigators who's working on that system is up next with Kelsey, who's going to talk about her work looking at these interactions specifically in the rhizosphere, or the areas around the roots of these plants. With that, I'll pass on to Kelsey.
All right, thank you, Peter, so much for that introduction. And so I'm really glad to be here today, and I'm just gonna dive right in. So soils store more carbon globally than the biosphere and atmosphere combined. And this is because plants are loading soils with carbon. And while much of this carbon is entering as the dead leaves and roots that are gradually broken down by fungi, bugs, and bacteria in the soil to form soil organic carbon, we often don't think about another important pathway by which plants deliver carbon to soils. And that's where living plants exude a complex mixture of compounds as well as soft cells into the surrounding soil environment in a process known as rhizodeposition. And these rhizodeposits have a complex makeup and perform a variety of functions in soils, including driving biogeochemical reactions and signaling between symbiotic and pathogenic microbes in a plant's rhizosphere. But you might be asking, Kelsey, rhizospheres? Well, rhizodeposits remain a pretty large question mark in our understanding of below-ground carbon cycling, both in terms of their production as well as their fate in soils and how they might be changing in response to future climate. After all, soil organic carbon may exist in soil anywhere from minutes if rapidly respired by microbes to millennia if stabilized through mechanisms such as mineral-organic associations. And as Peter mentioned, at the Belowground Biogeochemistry SFA, we're really interested in addressing some of these processes driving carbon cycling and looking at how they're impacted by warming and other climate drivers. And now we're starting to develop new experiments through both our Blodgett and Point Reyes field sites and deep soil warming experiments to look at the role of rhizodeposits in the future of carbon cycling. But to fully understand the complex factors affecting rhizodeposition, let's take a step back and recognize that almost all of the processes I've been talking about are examples of redox reactions or electron transfer reactions. And for many of us, redox is a concept that we might remember from our intro chemistry class, but it's actually one of the most important processes on the planet. It's happening right now in every single cell in your body, driving your metabolism. And the same is true for both microbes and plants. In fact, photosynthesis is another example of a redox reaction. And rhizodeposits themselves both drive redox reactions and are in turn shaped by redox conditions in soils. Redox is life. So as we dive into this question of how do rhizodeposits change in response to warming, not only are we looking at temperature as a driver, we're also looking at soil moisture and redox conditions at our field site continuously, alongside characterizing in situ rhizodeposit chemistry and trying to capture also the role of roots functionally in terms of production of rhizodeposits and microbes in processing that rhizodeposit carbon. And collectively, we hope that by trying to capture all of these different elements in our deep soil warming experiments, we might better understand the role of rhizodeposits in a warming world. So thank you very much.
Nicely done, Kelsey. Just as a friendly reminder, everyone, please remember to send reactions and congrats to the chat for all those awesome presenters. Are we ready for the first trivia questions? We'll be passing it off to Christine Buchner for that. Christine was a postdoc in the physical chemistry in the lab for about 3 years and is currently a program manager for the Belowground Biogeochemistry SFA. Christine, please take it away.
All right, everyone. Thanks, Mingfei. And yeah, keep those reactions coming. And let's get warmed up with our first trivia question. Should be pretty easy. I think someone's going to launch them. There we go. How many science focus areas will be housed in BioEPIC? Is it 4? Is it 6, 7, or how about 9? Make sure you get your vote in on the little pop-up on your screen. How many science focus areas are going to be in the BioEPIC building? All right, is it time to reveal the extremely surprising answer? It is going to be 4 scientific focus areas. Most people got that. I think some of us are just very optimistic about new and coming projects that maybe push up this number. So with this, I think it's time to talk about Enigma.
Awesome. Great. Well, thank you, Christine, and thank you, Kelsey. And thank you to everyone in the chat for sending your reactions and sending your congratulations to the chat. I know it's a little difficult in a virtual slam to make our speakers feel appreciated, but it goes a long way. Next up, we will be getting to know the Enigma SFA, which stands for Ecosystems and Networks Integrated with Genes and Molecular Assemblies. And if you take away nothing else from today, you should at least be impressed that our best and brightest scientists managed to whip up that acronym. We have here to talk about Enigma, Adam Arkin. Adam has one foot on the hill at Berkeley Lab as a senior faculty scientist in the Environmental Genomics and Systems Biology Division. He is the lead PI for DOE's Systems Biology Knowledge Base, or KBase, another fun acronym, and is also the scientific lead for the Enigma SFA. His other foot is down at the UC Berkeley campus in his role as a professor in the Department for Bioengineering and a lead for the Center for Utilization of Bioengineering in Space. Please welcome Adam Arkin.
Thanks, Jeremy. Right, so Enigma. So, so Enigma, and by the way, I'm going to blame Trent for that acronym, Trent Northern Special. We are interested in how microbes, molecule per molecule, assemble, interact with each other, and transform the terrestrial subsurface, in particular in mediating nitrate and nitrogen processes in that subsurface. And what I'm showing you here is kind of a map of where nitrate has become higher than EPA safety levels in the groundwater, and correlated with that is that it dissolves the shale and minerals to dissolve uranium and other heavy metals into that water. So each of these little red spots is a place where anthropogenically we've caused a problem like this. We work at Oak Ridge in this Y12 region where the Manhattan Project ran. And we're interested in how this contaminant plume from this waste storage pond is affecting the subsurface and how microbes adapt to that environment and can transform nitrate and uranium in various ways while they're down there. And to do that, we have this sort of multi-scale operations team. We're a very large group of people spanning a few national labs and universities. And we work together to bring together soil measurements, water measurements, chemistry, weather, and biology in time and space to understand how this occurs. And we bring that information back to lab to tear apart the organisms and how they work, again, gene per gene. So that's our general function, is to get a mechanistic and predictive model of how microbes are transforming the subsurface. And this hooks up to things that other people in BioEPIC are doing. So we're possibly the focusing on the lowest part of the system, from bedrock up to the variable saturated zone and maybe up into the beta zone a little bit, whereas other groups tend to be from the canopy of the plants down to the soil. so we can interface our processes with them quite a bit. I think with that, I can introduce our next speaker. Is that Josh? I think it must be Josh. So Josh Wong is a graduate student in comparative biochemistry, and he is working on one of the organisms that we can bring back from the field. This is an exemplar, a model member of one of those fields called Crutchfield-Peters mCAFEs. It's emblematic of organisms that can store carbon inside their bodies. And so he's interested in how they control, again, molecule per molecule, how they can control the storage of carbon and then also produce sustainable bioplastics. So Josh, take it away.
Thanks, Adam. Just like us humans saving money for financial hardships, bacteria also save resources for the tough times. I'm investigating how the bacteria Sinicator manages bioplastic carbon storage under different carbon nitrogen conditions. The bioplastic polyhydroxybutyrate, or PHB, is an important tool for Sinicator survival, a significant route of carbon sequestration in the ecosystem, and a decomposable material for industrial uses. As such, understanding the strategy of making them can help with both understanding the carbon cycle and improving the efficiency of biomanufacturing. Also, similar to us humans, individual bacteria have huge variations in their behavior, even with the same genetic composition. To resolve patterns within this colossal amount of variation and noise, we need to track down each of a large number of bacteria over a very long time. To achieve that, we built a microfluidic system that traps bacteria into tiny channels orthogonal to the nutrient flow to keep bacteria in constant conditions under a microscope. With this setup, we can track up to 100,000 bacteria at single cell resolution for up to 5 days. By reducing nitrogen concentration down to 10 to the minus 12 grams per liter, we found that contrary to common belief, Sinicator does not accelerate its PHB production at low nitrogen concentration. At exceedingly low nitrogen concentration near the top of the channel, cells stop growing but keep accumulating PHB, which increases not the production rate, but the PHB yield by improving PHB mass PHB mass per unit biomass, which we believe is what the others previously observed in bulk experiments. Sinicator actively controls its homeostasis over a huge range of nitrogen concentrations, keeping its PHB production rate constant over 1,000-fold and the growth rate constant across 12 orders of magnitude or trillion-fold of nitrogen. In order to improve production rates in an industrial setting, we need to break this control system first. A very small fraction, or about 2% of the cells, produce PHB at about twice the rate of the others. We believe it is a bad hedging strategy to give the population better resilience to extended periods of famine, with a few of them surviving significantly longer than the others. The strategies we discovered help Sinicator survive in the wild and sequester carbon from the atmosphere. The manipulation of these strategies can also help us produce more PHB towards a more decomposable future. Thanks a lot for listening.
Hey, it's my pleasure to introduce Jinwoo Im. He works with Michelle Newcomer over in ESA, and he is a specialist in mechanistic and AI-driven reactive transport models and has joined our project recently and is really changing the way that we integrate our data with a sort of a MODEX, model-driven experimentation approach. So Jinwoo, please take it away.
Thanks, Adam, for introducing me. I'd like to start my presentation by referring to human history, especially an early stage of human civilizations. In this time period, people built communities in a vast range of Earth environments, such as forest, desert, high plateau, and Arctic areas. As people adapted to each environment, they established their distinctive communities, developing different skills and cultures. And these ways of surviving feedback on the very Earth environment supporting us. Just like people, microbes are also forming different communities across vast, unique regions of the subsurface, rhizosphere zones, Vedox zones, variably saturated zones, and even contaminated zones. And as we do, microbes also impact their own environments, such as through acidification, weathering, clogging, and greenhouse gas emissions. Specifically, nitrous oxide emissions from subsurface are special because they represent more than 50% of the total global emissions. This is the topic I'm working on in the Enigma project. My work aims to understand when and how much nitrous oxide is emitted from subsurface in response to rainfall events, and what role subsurface microbial communities have on these emissions. In order to do that, I'm building a field-scale reactive transport model for nitrogen species in a subsurface. This field-scale model is coupled with an omics-level module that allows us to model microbial communities based on their genomic information. With this linkage across scales, the final model is able to show how different microbial communities emerge and evolve over hydrologic cycle in each region of the subsurface, and what functionalities they have contributes to this nitrous oxide emission. This cross-scale model becomes more realistic and scientifically meaningful when it is calibrated against field data. We have an intensive sampling site, which we call the subsurface observatory. We have collecting soils, water, and microbes and analyze them in laboratories. This large team science integrates into one mechanistic model across scales, which ultimately improve our understanding about environmental impacts on microbial communities and ecological impacts from microbial communities. Just as humans are building, constructing, and changing our own environments, so are microbes in a way that changes the very planet we lived on. Thanks for the attention.
Thanks so much, Josh and Jingwu. Great works. I'm super excited for those talks that you have you both just gave. So who wants to play some more trivia? This one will have Nikki Humphrey to present the question. Nikki has been the Deputy of Operations of the Biosciences area for almost 6 years. She is working to make sure BioEPIC operations are ready for the future occupants. Thanks so much, Nikki, and please take it away.
Thank you. Good morning, everyone. Our next question is, upon completion, how large will BioEPIC be? Is it A, 52,000 square feet, B, 67,000 square feet, C, 73,000 square feet, or D, 86,000 square feet? If you were listening closely to Susanna earlier, she gave you the answer. It looks like the results are in. I just lost them on my screen. There they are. And most of you got it right. The answer is C, 73,000 square feet. Thank you.
Thanks, Nikki. Now it is my pleasure to introduce Watershed Function SFA led by Owen Brody. Owen hails from Ireland and has made Berkeley Lab his home for a long lifetime. He is Deputy Director of ESA's Climate and Ecosystem Science Division and serves as an Associate Adjunct Professor in the Department of Environmental Science Policy and Management at UC Berkeley. Owen, please take it away.
Thank you, and good morning, everybody. Thanks for joining. I can't believe we're at the third BioEPIC Slam already. Well, first of all, it's my pleasure to be able to represent the Watershed Function SFA at this BioEPIC Slam. I'm coming to you virtually, of course, from the headwaters of the Colorado River, high up in the Rocky Mountains. And the Colorado River provides water for 40 million people across 7 US states. And as you might know, many of you might know, we're studying these systems because mountainous ecosystems are vulnerable to both current and future climate change, and particularly to changing snowpack and groundwater resources. And this has consequences for the quantity and the quality of water resources emanating from these important water towers of the world. To be able to build a predictive understanding of how these watersheds work, we need to consider their components or their traits. And these are parts of the watershed that interact, like the topography, the elevation, the hill slopes, the bedrock, the soils, the vegetation, the microbes, all of this together work in unison to influence the functions of the watershed and how much water and elements are exported from these watersheds. Now, to be able to quantify the flow of water and elements from pores to plateaus, we need to develop new sensing approaches. And that's where the new BioEPIC component of EcoSense comes in. a critical component to develop new sensing modalities to be able to tackle problems at this scale. Today, you'll hear from two of our innovative early career scientists, Stijn Wielandt and Nick Thiros, who are doing just that. And first of all, I'd like to introduce Stijn. Stijn is a research scientist who hails from a Belgian town called St. Nicholas. So, ho, 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 Stijn. And when he's not surfing mountain waves, Stijn likes to run. In fact, he's actually been known to chase movie stars. So next time you see him, you should ask him about it. So we're going to hear from Stijn now about what waves can tell us about water. And take it away, Stijn.
Thank you so much, Owen. So yeah, in the Watershed Function SFA, a large team of scientists investigates basically all aspects of water cycles, and that's from the groundwater below our feet to the clouds and everything in between. And so me being an engineer, I work on technologies that can give scientists more accurate information about these water-related processes. And so my work focuses on radio waves. And so basically, what are radio waves? Actually, it doesn't matter. Let's just talk about sound waves instead, because at least we can hear those. And so if we think about sound waves, there's two things we can use them for. Of course, we can use sounds to communicate, which I'll talk about in a minute, but we can also use sound waves to sense or measure things. And so, for example, we can think about bats. Bats use high-frequency sound waves for echolocation to find their prey. And we can actually use exactly the same technique to measure radio wave echoes bouncing off snowpack to measure its thickness. There's also another thing we can measure with waves. We all know that you can measure how far you are from a lightning strike by counting the time until the thunder. And that's because the sound waves travel at a certain speed. And now this speed is different when the waves travels through air as opposed to ice, for example. And so we use this phenomenon to measure the amount of ice in the snowpack, and thus also the amount of water that is contained in the snowpack. And so scientists, they can use this to assess the amount of water that is actually going to be released during snowmelt. So that's another thing we measure with sounds or waves. And so another sound effect that we've all observed is when an ambulance drives by. So when an ambulance drives by, you hear the pitch of the siren that seems to be higher when it approaches and then lower after it has passed. And so we call this a Doppler effect, and we can actually use this to measure the speed of raindrops or snowflakes or even hail falling down. And that is because the pitch of the reflection or the reflections or the echoes of these particles, it depends on their speed. And so also the more intense the precipitation, the louder we can actually hear those echoes. And now, as I said, we can use sound waves not only for measuring things, but we can also use it for communicating. When we look at whales, for example, we can see that they communicate at very low frequencies. And these low frequency communications, they can travel very far, and it takes only a very little energy. So that is great when we have these remote sensors in the field that just work on batteries. And so the only disadvantage about this is that whales, they talk very slowly, so we cannot send that much data at these low frequencies. So that's why we also have systems that communicate at very high frequencies, basically just like chipmunks. These sounds you can only hear when you're nearby, but just like chipmunks, you can talk very fast and you can send a lot of data very quickly. So basically, when we build these sensors that measure water-related parameters with waves, we also want to communicate with them using waves so that they can send their data in real time to our BioEPIC facility. And then there, supercomputers and scientists, they can take control and analyze these data in real time and actually create self-driving field laboratories. Thank you.
Thank you, Stijn. So creative. Thank you. Well, now it's my great pleasure to introduce Nick Thiros. Nick is an exceptional postdoc who searches for the fingerprints of climate change in pretty unlikely places. Nick hails from Salt Lake City in Utah. Nick loves to run, to ski, and to bike. And today he's going to tell us about the world's water towers, fingerprinting flows from snow to society. All yours, Nick.
All right, thank you, Owen. So have you ever been out in the mountains on a real hot summer day when there's no more snow to melt and it hasn't rained for weeks? And have you wondered, how is there still so much water running in the streams and rivers? Well, this sustained stream flow is largely due to the slow release of groundwater that is actually stored within the soils and bedrock beneath our feet. And understanding the groundwater system is really especially important in the Western United States, given that the mountain systems here are the major source of water for downstream users. And because of this, I, as well as many others, not only see mountains as these picturesque playgrounds to go run, ski, and bike in, but also as crucial water towers that support our linked food, energy, and water resources. So there are some very important questions that we need to better understand about these mountain groundwater systems, though. For instance, how much groundwater is actually stored in these water towers? How fast do these water towers fill up and drain each year? And can these water towers actually support stream flow and ecologic function during multi-year drought conditions? But despite this importance of the groundwater system, it still remains quite uncertain. And this is largely due to the big challenges in directly observing the subsurface in these steep and really challenging to access mountainous environments. So within the Watershed Function SFA, my research directly investigates some of these groundwater related questions. And a real key observation data set that I use are geochemical tracers that are found within the groundwater. These tracers can be used as a real fingerprint to understand hydrologic and groundwater processes. For instance, how fast is the groundwater and how deep is it flowing within the mountain? Where does snowmelt or rain first enter the system and become groundwater? And what zones does the groundwater flow through? These tracers that I measure are really particularly useful to constrain how long the groundwater has been in the system, which is what we call groundwater age. For instance, groundwater that moves really fast will have very young ages and is expected to dictate hydrobiogeochemistry different than slow-moving groundwater. And quite surprisingly, in the East River catchment, we find groundwater can be on the order of hundreds to potentially thousands of years old. And we really need to understand what are the watershed traits that are actually controlling this response that we're seeing. And further within the Watershed Function SFA, we have begun to tackle this challenge of integrated groundwater, surface water predictions, especially under perturbed climate and warming using numerical models. Our models actually show that groundwater is declining with warming, which can have consequences on downstream water quality in addition to quantity, as well as some biogeochemical cycling. So thanks again for listening, and I hope these insights on mountain groundwater will be very useful for all of us.
Thank you so much, Nicholas and Stan. Those were some excellent talks. It's really fascinating to hear both about the role of radio waves in understanding our water resources and kind of what's happening beneath the surface to make those water resources the water towers that they are. This next round of trivia is being led by Astrid Terry, who is the project manager for both the mCAFEs and Enigma SFAs. Astrid?
Thank you. So this question is, what is the overall research focus that all BioEPIC tenants have in common? A, how fungi can help improve water, food, and energy systems. B, how microbes can improve atmospheric carbon storage. C, microbial communities and how they interact with cells and bacteria. D, microbial communities and how they interact with the environment. And pretty much everyone got it. The answer is D, microbial communities and how they interact with the environment. Well done.
Thanks, Astrid. Now, last but certainly not least, to introduce mCAFEs, which is acronym of Microbial Community Analysis and Functional Evaluation in Soils, we have Trent Northern. Among the many hats Trent wears, he serves as Senior Scientist and Deputy Director of the Environmental Genomics and Systems Biology Division. He is also the Director of High Throughput Biochemistry of the Joint Bioenergy Institute and Metabolomics Program Lead at the Joint Genome Institute. Relevant to today's event, Trent served as a Laboratory Research Manager for mCAFEs, where they are trying to understand and control micro-plant-root interactions to support sustainable bioenergy. Take it away, Trent.
All right, thank you, Mingfei. Well, it's my pleasure to be here today and tell you about this diverse project that we're working on called Microbial Community Analysis and Functional Evaluation in Soils. And as Kelsey introduced, the rhizosphere is really a critical zone for addressing DOE emissions. So this is the region of soil that's just immediately around the plant roots. And as Kelsey mentioned, a large fraction of the carbon that enters soils is coming from plants. They pump up to about 30% of the carbon that they fix below ground, where it is thought to benefit microorganisms that in turn benefit the plant health. And since this is such a large fraction of the carbon that enters the soil, it's critical for understanding the global carbon cycle. And these microbes that are benefiting the plant can reduce the amount of chemical fertilizers that we need. So this is very important for sustainable agriculture. But unfortunately, there are many challenges to understanding these communities. They're very complex. Most of the microbes are uncultivated. And so we don't know very much about their genes and their interaction. And so what mCAFEs is doing is developing in-depth genetic and biochemical characterization capabilities and combining this with spatial temporal modeling and critically in situ community editing technologies within fabricated ecosystems. So let's just say a few more words about that. So mCAFEs can test the function of an uncultivated organism directly within a community to understand genes that mediate key environmental processes, including interacting with the plant host. And to do this, we use a whole suite of fabricated ecosystems, from small scale single plant devices we call ecofab to bigger systems, which are known as ecopods. And these are capabilities that are going into the BioEPIC building. And today I'm delighted to introduce two really talented early career scientists that will be telling you a little bit more about our science. First, Veronica, who is a postdoctoral researcher in the Del Nino lab at LBNL, will tell you about aromatic acids in the spotlight, unraveling their influence on plant microbe interaction. And next you'll hear from Ilya after that. So I will turn it over to Veronica.
Okay, can everyone hear me? Okay. Well, thank you for that introduction, Trent. So hi everyone, my name is Veronica, and I'm a microbiologist in the mCAFEs SFA. And today I want to talk to you about the dynamic interactions that happen between plants and microbes that live in the rhizosphere, specifically bacteria. Now the rhizosphere, just like Trent mentioned, is the soil just around the roots. But what's amazing about it is that it's home to millions of microorganisms that we know as the microbiome. This can house fungi, phages, and my personal favorite, bacteria. Amongst them, bacteria play an important role in supporting plant health by providing them with nutrients like nitrogen and hormones and protecting against pathogens. But how do plants communicate with these microbes? Well, they release what are known as root exudates. These root exudates are metabolites that can act as signals to attract specific bacteria to satisfy the plant's needs. But there's so many of them, so my research focuses on a group of them, known as aromatic acids, which are widely produced by plants, but we still don't understand how they can exactly change the way bacteria interact with plants. In particular, I am interested in how bacteria that are attracted to them might respond to them and use them. My research hones on one particular bacterium at the moment, known as Berkeley. What's interesting about it is that it's able to thrive around these aromatic acids and is able to eat them and use them as a nutrient source. So using resources from the mCAFEs SFA team, like this collection of mutants on Berkeley, which carry single gene mutations, we can uncover how it does this and ultimately test if blocking the ability at the genetic level of eating these metabolites can impact the ability of bacteria to thrive in the rhizosphere of a plant that produces aromatic acids. or if perhaps other bacterial genes are more important. Ultimately, but how do we translate these findings that we see in the lab to more real-world scenarios like the field? That's where the EcoPod comes in. This large chamber that will be housed in the BioEPIC building allows us to simulate natural environments like those in the field and test how these interactions that we see in the lab can change in a more natural setting. So, in conclusion, the rhizosphere is the dynamic stage where complex communications between plants and microbes happen. By dissecting these interactions like we hold the key to, we hold the potential of using microbial communities for a more sustainable bioenergy solutions. Thank you.
Perfect timings. That was great, Veronica. So now it's my pleasure to introduce Ilya Dukovsky, who's a research assistant professor working with Daniel Segre, and he's going to be telling us about rooting for models, metabolic modeling of the rhizosphere microbiome. Okay, take it away, Ilya.
Thank you, Trent. Let's take a look at a potted plant over here. You would say that there is only one organism present in that pot. Well, that's not true. There's a lot of microorganisms like bacteria that are living on the root and in the area around the root. We call that space the rhizosphere. The community of microorganisms that are living in the rhizosphere, we call them rhizosphere microbiome. There's lots of activities going on in the microbiome. The bacteria are eating food, they're growing, they're replicating, and they're producing waste that they want to get rid of. Now, what's waste of one bacterium is the treasure or food of another one. So the bacteria are trading these foods in a friendly, mutually beneficial way. Sometimes they compete if they happen to like the same kind of food. This creates a complex network of interactions in the microbiome. We want to study these interactions to understand if all that activity in the microbiome is beneficial for the plants or maybe it's harmful. We do that by using this transparent pot-like device we called EcoFab, with which we can take a good look at the root system and study it, sample it, and study it. But we also want to create models of the microbiome shown here and the EcoFab if our models can reproduce some of the experimental results, or better yet, if we can make predictions and then confirm those predictions in the experiments, we will know that we are on the right path for understanding the complexity of the rhizosphere microbiome. How do we do the modeling? We start by reading the DNA of the organisms. And over here, we start by reading the DNA of the organisms. And from that, we figure out all the chemicals reactions that are present in the cell. Then we use, we create a model of metabolism of the organism, and we use a computational algorithm that will tell us the rate that the bacteria are eating the food, growing, and secreting the waste. With that, we can create a simulation of the complex network of interactions in the microbiome. One of the advantages of the modeling is that we can easily take out an organism from the system or add a new one, just like a video game where you can easily kill a character or introduce a new one. So that's how we can simulate the system, and these changes in the system will affect the plant. And from our simulations, we can help experimentalists decide what to do next to make the plant grow better and stronger. Thank you.
Great job, Veronica and Ilya. Those talks are fantastic and amazing works from all of those presenters for this BioEPIC Slam. So to recap it all, we heard from the Belowground Biogeochemistry SFA with Kelsey, and Enigma represented by Josh and Jing Wu. And for the Watershed Function, we have Stijn and Nick. And for last but not least, at the mCAFEs, we have Veronica and Ilya.
And now the time has finally come to vote and decide the winner of this year's slam. So I encourage all of the presenters to join us and turn on their cameras. And audience, first, let's show your appreciation and share your reactions and your words of encouragement for all of these wonderful talks that we've seen today. And the link to vote for your finalist, or rather your champion, should have just been put in the chat from Lida Gifford. So you have 2 minutes to vote using this link. Go ahead and do it now. Be as quick as possible. In the meantime, while all of you are voting, we've got one more trivia treat for you. This time it's hosted by Orlando Cassiano, who is ESA's very own Director of Operations, and who's been working tirelessly to get BioEPIC operations ready for the future occupants. Orlando, while everyone's voting, would you be willing to hop on and give us that last trivia question?
Absolutely. Okay, so last but not least is mine. So here we go. Of the following technologies that will be featured at BioEPIC, which is not a fabricated ecosystem? Is it A, EcoPod, B, Smart Soils Testbed, C, EcoSim, or is it D, EcoFab? Again, which is not a fabricated ecosystem. And yes, we got it. It is C, EcoSim. Pretty close there. Awesome.
Nicely done. Everyone was completely on it today with the trivia. Thank you so much, Orlando, for popping in with that last question. Voting should be wrapping up now. If you haven't already voted, get in there. It sounded like a couple people might have been having trouble with the link, but I think this most recent one, go.lbl.gov/bioepic24 vote, should be good. I'll just go ahead and put an executive timer on it and say we've got 10 more seconds to vote. Go 5, 4, 3, 2, 1. All right. With that, voting is coming to a close, and I've got some helpers in the background who will be tallying the votes.
Wasn't that incredible? It was super fun to hear about all the research that will take place in our newest building. Congratulations to each of the participants for their engaging talks. I really enjoyed each one of them. I'm pretty sure you all struggle picking a winner. With the level of talent on display today, I am pretty excited to see how the BioEPIC facility will bring it all together under one roof and further advance the already fruitful collaboration between biosciences and earth environmental sciences at Berkeley Lab.
Totally agreed. Yeah, it was really awesome to hear all these talks. And after all these years of seeing the building itself slowly kind of rise from the ground over by Building 91, it's been awesome to see it nearing completion and contemplate what it's going to be like for us to move in later this year and next year. So beyond just providing a home for our scientists, I'm also really excited that the advanced facilities in the BioEPIC building are going to enable entirely new types of science from researchers like the ones that you just heard from today. And hearing all these talks and seeing all these like really astute and interested and passionate scientists, I'm just very sure that we're in good hands. And I'm really looking forward to seeing what we can make happen with this new building. Yeah, I think we've got our winners. So Mingfei, do you want to announce the first?
Yeah. So without further delay, let's announce the winner of our first honorable mention award, who will get a spot award for $100. So the honorable mention go to Stijn Wielandt, who is from the Watershed Function SFA, and the title and talk about sorry, I cannot put the I forgot which you did an amazing job. Sorry, I was a bit nervous. Yeah, great job.
Thanks so much. Thank you.
Amazing. And with that, it's my pleasure to announce our first runner-up, who is going to receive a spot award for $175. And that goes to Veronica Escalante. from mCAFEs.
So
and that leaves Mingfei. Yeah, go for it.
So last but not least, let's welcome the next and the newest BioEPIC Research Slam Champion, who will get a spot award for $250 and with the bragging rights, of course. We have Kelsey from the Belowground Biogeochemistry SFA. Nice work, Kelsey. Great job.
Thank you. Thank you very much. Thanks for organizing this. It was super fun.
Woo! Why is so serious? Amazing. Well, congratulations again to our BioEPIC champ, Kelsey. I think everyone agrees that was well-deserved, but also that there was some very tough competition.
Yeah, I truly enjoyed everyone's talk. Amazing job, everyone. You are all the winners in my heart. Thank you so much, Jeremy, for being my co-host for this exciting event.
Yeah, and thank you, Mingfei. It was really a pleasure to host along a real BioEPIC Slam champion of old. And awesome to see how much the folks from this year really gave you a run for your money. I think the competition's getting stiffer every year. It's good that we're wrapping it up this year, because otherwise I don't know how anyone would pick.
Yeah. Thanks everyone for coming today, and you can follow our upcoming events on social media at #BioEPIC, and hope you enjoy the rest of your day. And thanks everyone for coming and for all those amazing presentations. Hope to see you soon. Bye.